The Honourable Mr. K.K. Chan, Chief Secretary for Administration, the Government of the Hong Kong Special Administrative Region. Dr. the Honourable Christine Choi, Secretary for Education of the Government of the Hong Kong SAR. Our Honourable Laureates and accompanying persons, forum participants, distinguished guests, ladies and gentlemen, and our friends from the media, good morning. On behalf of the Council of the Hong Kong Laureate Forum, also known as the HKLF, a very warm welcome to the grand opening ceremony of the Hong Kong Laureate Forum 2025. This is Patricia Yang, and it's my pleasure hosting for you this morning. Let's put our hands together to welcome everybody joining us this morning. Thank you. It is truly a privilege to witness such a remarkable crowd and gathering of brilliant minds here at the Hong Kong Science Park, a venue that exemplifies Hong Kong's commitment to innovation and scientific excellence. Over the coming days, we're honored to have 12 Shau laureates, together with distinguished scientists, interact with over 200 foreign participants from different parts of the world in this cross-generational, educational, and scientific exchange event. As we commence this year's forum, we're honored by the presence of many individuals and organizations who have played instrumental roles in bringing this event to fruition. At this juncture, we'd like to take this opportunity to extend our heartfelt gratitude to the government of the Hong Kong Special Administrative Region, our principal sponsor, the Li Shao Key Foundation, which fully sponsors the Hong Kong Laureate Forum, our major partner, the Shaw Prize Foundation, members of the Scientific Review Panel and Program Steering Committees, local academic partners and collaborating organizations, not to mention our participating Shaw laureates, their accompanying persons, and local speakers who take time from their busy schedules to join us. May I ask all of you to put your hands together once again to thank them for us. Thank you very much. Now, to officially begin, may we first invite Professor Timothy Tong, Chairman of the Council of the Hong Kong Laureate Forum, to give us a welcome speech. Please welcome Professor Tong.
The Honourable Mr. K.K. Chan, Chief Secretary for Administration, who is also acting Chief Executive today. Dr. the Honourable Christine Choi, Secretary for Education, esteemed Shaw laureates, fellow council members, distinguished guests, young scientists, members of the press, ladies and gentlemen, good morning. Let me take this opportunity to recognize the two government officials who have so kindly joined us this morning. The Chief Secretary for Administration, Mr. K.K. Chan, will be our officiating guest. Let us give Mr. Chan a round of applause. Thank you. And Dr. the Honourable Christine Choi, Secretary for Education, special friend of ours. Thank you for coming. Let's give Christine a round of applause too. Thank you. Thank you. It is indeed my honor to welcome all of you to this opening session of the second Hong Kong Laureate Forum. This is a special gathering centered on nurturing future generations of scientists through connecting scientists across generations, cultures, and disciplines. It is a dynamic platform for inspiring young or aspiring scientists to deepen their interests in science and maybe to pursue doing scientific research as a career in the future. Over the coming days, participants will engage in keynote lectures, thematic discussions, laboratory visits, and cultural activities designed to promote exchange of ideas and spark collaborations. Through dialogues and meaningful networking, we aim to cultivate curiosity, and ignite passion for science among the younger generations, both in Hong Kong and in the international community. This year, we are honored to host a distinguished group of Shau laureates whose groundbreaking contributions exemplify the highest standards of scientific excellence. Alongside them are hundreds of young scientists from Hong Kong and abroad, whose fresh perspective and enthusiasm are vital to shaping the future of the research world. To all of you, Shau laureates and young scientists alike, your presence and active participation are what make this forum truly impactful. We hope this experience will inspire you, challenge you, and connect you in ways that extend far beyond these few days. And of course, none of this would be possible without the steadfast support of our partners and the wider community. We extend our sincere gratitude to our principal sponsor, the Li Shaoqi Foundation, whose generous financial support and enduring commitment to education, youth development, and scientific advancement have been instrumental in making the Hong Kong Laureate Forum a reality. We are also deeply thankful to our major partner, the Shaw Prize Foundation, whose recognition of outstanding achievements in science forms the very foundation of this forum. The Shau laureates' engagement with young scientists brings our mission to life. We also wish to thank the Hong Kong SAR government, our board members, and our academic and industry collaborators for their invaluable contributions and unwavering belief in the power of science. And now, it is my privilege to invite our officiating guest to deliver his address. His presence today underscores the government's strong commitment to nurturing scientific talent and advancing Hong Kong's role as a global hub for innovation and technology. Ladies and gentlemen, please join me in welcoming the Honorable Mr. K.K. Chan, Chief Secretary for Administration of the Government of the Hong Kong Special Administrative Region. Thank you.
Professor Timothy Tong, Dr. Colin Nam, Dr. Raymond Chen, Shau laureates, young scientists, ladies and gentlemen, good morning. It is a great pleasure to join you today for the opening ceremony of the Hong Kong Laureate Forum, the second edition. I'm delighted and honored to welcome the distinguished Shau laureates together with 200 exceptional young scientists from Hong Kong and from around the world. As Professor Tong noted, this forum was created to support science globally and locally, and to fast-track Hong Kong's rise as an innovation and technology hub. Today's venue, the Hong Kong Science Park, our flagship INT center, perfectly reflects that ambition. Hong Kong is fast emerging as an international INT center. In September, the Shenzhen, Hong Kong, Guangzhou Innovation Cluster topped the Global Innovation Index ranking of the world's top 100 innovation clusters. Talent drives technology. Hong Kong recognizes the important role young scientists play in advancing science and technology, which is why we invited some 200 of you here for this year's four-day forum. It's also why our government has developed a series of policies to attract strategic talent to Hong Kong. And the results are encouraging. Hong Kong ranked first in Asia and fourth in the world in this year's World Talent Ranking, and we topped the world in the Graduates in Science category. In September, we launched the 3 billion Hong Kong dollars, approximately 386 million US dollars, Frontier Technology Research Support Scheme. It is designed to help our 8 publicly funded universities recruit top scientists and researchers to Hong Kong and to build the facilities they need to conduct breakthrough research. Hong Kong is home to 5 universities that rank among the global top 100. They are producing cutting-edge research and training the next generation scientists and innovators. We are also establishing an advanced INT system integrating 3 major INT parks and 5 key research and development institutes. We are doing so under the principle of promoting technology with talent, leading industries with technology, and attracting talent with industries. The goal is to grow our talent pool, boost innovation, and support our country's science and technology development. This forum has a packed schedule. Our young scientists can look forward to panel discussions, a public forum, and, for some, a day trip to Shenzhen. Shau laureates will be speaking at many of these events. And, of course, the many chances to meet and exchange ideas will be equally rewarding. I have no doubts that the forum and all those taking part in it will spark fresh insights and new collaborations. For that, I'm grateful to the organizer, the Council of the Hong Kong Laureate Forum. My thanks also go to the principal sponsor, the Li Xiaoqi Foundation, and to the forum's major partner, the Shaw Prize Foundation. Science needs brilliant minds, and our society needs engaged citizens. For those of you from Hong Kong, I must encourage you to vote at the December 7th Legislative Council election and bring your delegation from the laboratory to the ballot box. Ladies and gentlemen, I wish you all the best in your scientific work and a memorable week here in Hong Kong. Our city is also a rising East-West Center for international cultural exchange. From the West Kowloon Cultural Center, one of the world's largest art and culture developments, to our beautiful country parks, our famous food, and our vibrant nightlife. The good life is all around you. Enjoy it all. Thank you.
Thank you. Thank you very much, Chief Secretary. Please be seated. And on behalf of the HKLF, we express our heartfelt gratitude to the Li Xiaoqi Foundation for the generous support and unwavering support, not only financially, but also through invaluable operational facilitation. Beyond its contribution to the HKLF, the foundation has long been dedicated to nurturing young talents and promoting educational advancement, thereby creating a positive and lasting impact on society. And now, ladies and gentlemen, please join me in welcoming Dr. Colin Lam, Vice Chairman of Henderson Land Group and Director of Li Xiaoqi Foundation, to share a few words. Dr. Lam, please.
The Honorable Mr. K.K. Chan, Chief Secretary for Administration, Dr. the Honorable Christine Chow, Ms. Choi, Secretary for Education, esteemed Shaw laureates, fellow council members, distinguished guests, young scientists, ladies and gentlemen. Good morning. It is my tremendous pleasure to join you today representing the Li Xiaoqi Foundation. It is truly inspiring to be in the company of so many distinguished Shaw laureates and bright young scientists from around the world. The Li Xiaoqi Foundation is delighted to be the principal sponsor again of this forum. Our late founder, Dr. Li Xiaoqi, saw education as the most honorable investment that can reap far-reaching and immeasurable returns for the society. The Foundation and the Henderson Land Group have supported education throughout the decades, both in Hong Kong and around the world, sowing the seeds of learning for future generations. Our approach echoes the Greek proverb, "A society grows when the old men plant trees under which they never sit." Likewise, in Chinese, it is 前人種樹，後人乘涼. We are proud to be part of the mission to foster cross-generational dialogues and to light a fire of inquiries in young hearts. The ingenuity of vision of young scientists can harness the great potential of technologies and shape a better world for the future. On behalf of the Li Shaoqi Foundation, I'd like to express our sincere gratitude to the Council of the Hong Kong Laureate Forum, chaired by Professor Tang, to the Shaw Prize Foundation, and to the Hong Kong SAR government for their unwavering support and partnership. I wish this forum a great success. Thank you very much.
Thank you. Thank you, Dr. Lam. Please take your seat. At the HKLF, we're also very privileged to have the Shaw Prize Foundation as our major partner. The Shaw Prize Foundation organizes the internationally acclaimed Shaw Prize, which recognizes scientists who have made exceptional contributions to scientific research and its applications. Now, may we invite on stage Dr. Raymond Chan, Chair of the Shaw Prize Foundation, to share a few words. Let's welcome Dr. Chan.
The Honorable Mr. K.K. Chan, Dr. Christine Choi, esteemed Shaw laureates, young scientists, good morning. 2 weeks ago, we gathered at the Shaw Prize Award presentation ceremony to celebrate 4 leading international scientists who exemplify the spirit of human curiosity, endeavor, and achievement. We are very pleased to have our laureate in Life Science and Medicine 2025, Professor Wolfgang Baumeister, here with us today. If the Shaw Prize represents one summit of success, then this Hong Kong Laureate Forum can be seen as a base camp, a platform from which the next great expeditions to scientific discovery embark, a place intended for seasoned navigators to collaborate with and mentor young explorers to chart new frontiers, exchange ideas, and inspire one another. The Shaw Prize Foundation is deeply honored to be a major partner on this significant journey. The Hong Kong Laureate Forum and we share one firm belief: scientific research is rarely a solitary pursuit, but more often a collective journey, and sometimes a relay across generations. The Shaw Prize recognizes those who have so nobly carried the torch. The forum ensures this torch is passed to capable hands, and that its light continues to shine ever brighter. This partnership is a natural extension of our mission. Now in our 3rd decade, the Shaw Prize celebrates excellence in astronomy, life science and medicine, and mathematical sciences. Our founder, the late Sir Raymond Shaw, believed that excellence is not only measured by achievement and success, but by the act of inspiring others and giving back. He channeled his success into helping others and nurturing those who will shape the future. The Hong Kong Laureate Forum provides a unique platform to do the same. It brings together Shaw laureates and young scientists into an environment which promotes open dialogue, mentorship, and free exchange of ideas. A space where curiosity is encouraged, questions are welcomed, and new perspectives are born. It also reflects Hong Kong's growing role as a vibrant hub for global scientific discourse and innovation. Together, we hope to use the pursuit of knowledge to continue to build bridges between generations, between disciplines, and between people. Thank you.
Thank you. Thank you, Dr. Chan. Please be seated. In just a few moments, we'll officially launch the Hong Kong Laureate Forum 2025 and mark the beginning of this 4-day program filled with inspiring discussions, groundbreaking ideas, and enriching exchanges among our 12 laureates, distinguished guests, and young scientists. From today to the 8th of November, Shaw laureates and distinguished scientists will engage with over 200 forum participants from around the world, sharing insights, experiences, and aspirations across a wide range of scientific disciplines. And through world-class seminars, workshops, discussion groups, and poster sessions, visits to Hong Kong's leading research centers and the Greater Bay Area, participants will explore new frontiers of knowledge and foster cross-cultural collaboration with the global scientific community. And now, may we have the honor to invite our officiating guests to join us on stage and conduct the opening ceremony. First, let's welcome the Honorable Mr. K.K. Chan, Chief Secretary for Administration of the Government of the Hong Kong SAR. Next, please welcome Professor Timothy Tong, Chairman of the Council of the Hong Kong Laureate Forum. Dr. Colin Lam, the Vice Chairman of Henderson Land Group and Director of Li Shao Key Foundation. Dr. Raymond Chan, Chair of the Shaw Prize Foundation. Let's welcome them to the stage. The kickoff podium in front of us symbolizes the unification of inspirational minds from around the world. And now, our officiating guests will be holding the keys in their hands for the first photo. Now, would our officiating guests please hold the keys, and we'll take the first photo. Please smile for the camera at the front. The keys represent the power of knowledge, the spirit of collaboration, and our shared pursuit of discovery. And later on my count to 1, I'd like to invite our officiating guests to pick up the keys and have them ready right above the positions on the podium in front of you. And you place the keys into the podium that will illuminate the bridge where science transcends generations and guides us towards a brighter and united future. And now, would all guests please pick up the keys and have them ready, pointing above the space in front of you. And let's do the count together. Ready? 3, 2, 1. The Hong Kong Laureate Forum 2025 is now officially open. We look forward to exploring the world through the eyes of our distinguished scientists whose insights will inspire and illuminate the path ahead. Well, would our officiating guests please smile for the cameras for another photo? A truly amazing moment to remember. We also have our cameras for our media friends. So right after this photo, I'd like to direct the attention of our officiating guests to the back for our media friends' cameras. And would our officiating guests please continue to remain on stage for the following photos. Next, we would like to invite the Honorable Dr. Christine Choi, Secretary for Education of the Government of the Hong Kong SAR, and our board member of the Council of the Hong Kong Laureate Forum, Professor Kenneth Young, to join us for the photo. We're truly honored to have So many distinguished guests with us today, and let's get ready for this photo. Please smile for the cameras at the front. Please hold for one more photo. Our media friends are going to capture this moment as well. Now with our officiating guests, please smile for the cameras at the back. Please remain on stage. Now we'd also like to invite our Honorable Shau laureates participating at the forum to join us at this photo. Please welcome Professor Wolfgang Baumeister. Let's put our hands together to welcome our distinguished Shau laureates. Next, please welcome Professor Michael Perryman. Professor Matthew Bales. Professor Nigel Hitchen. Professor Simon White. Professor John Peacock. Professor Peter Water. Professor George Lustig. Professor Katsutoshi Mori. And Professor Stephen Balbus. We're truly honored to have all our Shau laureates with us at the forum. Let's have this photo together. Please smile for the cameras at the front. Kindly make a few steps towards the center. Perfect. Big smiles, and that will also mark the beginning of our 4-day Hong Kong Laureate Forum 2025. Okay, please hold. We have our media friends at the back so that they can capture this moment as well, please. All right, ladies and gentlemen, let's put our hands together to thank all our guests on stage. Thank you very much. Would all guests please proceed off stage and be seated? Thank you. I'm sure all of us are very excited to hear from our distinguished scientists joining us at the forum and exchange a lot of ideas to explore the world of unlimited possibilities. Now, ladies and gentlemen, at this moment, we would like to have all of you to participate because we're going to take a grand group photo with every one of you joining us today. Our photographers will be coming up to this stage to capture this moment for us, so please stay seated and have your smiles ready for this photo. All right, our photographers are ready at the center of the stage. Big smiles. We'll take a few photos. Excellent. And now, in just a few moments, our video camera will also capture this moment for us. So if you may, just wave at the camera at the stage on my count to 1. We're going to have a fabulous video capturing this moment. All right, ready? 3, 2, 1, go! Fantastic. Thank you very much, everyone. Thank you. Thank you for your participation. Thank you so much. Ladies and gentlemen, at this juncture, Chief Secretary, the Honorable Mr. Chan, and Secretary, the Honorable Dr. Choi, will have to depart now because of official duties. May I invite all of you to put your hands together to once again thank them for their presence today. Thank you very much. Thank you again. Now, ladies and gentlemen, we'll move on to the next part of our program, the keynote speech presented by Professor Wolfgang Baumeister, 2025 Shau Laureate in Life Science and Medicine. Before we invite him to the stage, I would like to provide you with a brief background information. Professor Baumeister is internationally recognized for his pioneering development and use of cryogenic electron tomography. This advanced imaging technique allows scientists to visualize three-dimensional structures of biological samples, including proteins, macromolecular complexes, and cellular compartments as they exist in their natural cellular settings. His research has developed and applied methods to reveal the inner workings of cells at an unprecedented near atomic level. The power of this technology is transforming our understanding of normal life processes and how they go awry in disease. And today, he's going to share his insights on structural biology in situ, state of the art, and prospects for the future. The following session will offer us a glimpse into both the remarkable achievements of both biotechnology, modern biology, and the exciting possibilities ahead. Now, ladies and gentlemen, please join me in welcoming Professor Baumeister.
Yeah, good morning. First of all, I wish to apologize for giving my lecture sitting, but I have a pretty nasty back problem and standing is quite painful. So, yes, I will talk about structural biology in situ. Let me begin by briefly explaining why we want to do structural biology in situ. I think traditionally structural biologists approach cellular complexity in a reductionist manner. So cells are taken apart, the molecular inventory is released, fractionated, and one or the other molecule is purified and then studied by the existing methods of structural biology, X-ray crystallography, increasingly cryo-electron microscopy, single particle analysis, or NMR. But it's also fair to say that rarely cellular functions are performed by individual molecules. Cellular functions arise from the interactions between the many molecular species inhabiting cells, which have to act somehow in concert. So the traditional approach, has been very successful if you look at the protein data bank. But nevertheless, I think there is a need to be able to study macromolecular, supramolecular structures in their functional environment. The functional environment is, after all, an unperturbed cell. So maybe the monitor could be turned a little bit so that it's readable. So I think the method that enables doing structural biology in situ is cryo-electron tomography. What is that? Very briefly, it's the application of tomographic principles of data acquisition. We have to take images from various different directions and then merge them into a three-dimensional structure by procedures such as weighted back projection. Now, so by doing that, we combine the power of high-resolution three-dimensional imaging with the best structural preservation that is physically possible to achieve. I think the goal of in situ structural studies must be that each and every macromolecule or each and every organelle is strictly kept at its native place. So what are the problems? Why has been cryo-electron tomography developed only over the past, 20 or 30 years? I think the problem is basically that to obtain a detailed and undistorted reconstruction, a tilt series must be recorded that covers as wide an angular range as possible with many increments as possible, as you see here in this little simulation. So going to 72 projections instead of 18, very clearly improves the quality of the reconstruction. So we would like to maximize the number of projections, but at the same time, we have to minimize exposure to the electron beam. And that is obviously a challenge, and this challenge can only be resolved by the solution is dose fractionation, basically distributing the, let's say, allowable electron dose over as many projections as possible. That the consequence is, of course, that each of the projections is heavily underexposed, and we can do it only to the extent that we are still able to align the projection images by cross-correlation. We need a well-defined cross-correlation peak. That requires some very serious automation of the data acquisition with a lot of auto-tuning functions. And that was difficult to do, let's say, 20, 30 years ago when electron microscopes were designed for manual operation and not easily amenable to automation. Now, historically, that is the first cryo-electron tomogram ever published. It's something as trivial and boring as lipid vesicles embedded in ice. Now, because of the dose limitations, the reconstructions in tomography are pretty noisy, so we began at that time to develop so-called denoising procedures, reducing the noise level while maintaining, not affecting the signal. Now, from lipid vesicles to something as small as a virus particle, in this case herpes simplex virus particle, that was a relatively small step. And so what you see here is a tomogram on the bottom, on the left-hand side of the entire virus. On the right-hand side, a cut-open view. We see in blue the nucleocapsid. Now, this looks, of course, already pretty much well improved in signal-to-noise ratio, because whenever in a tomogram there's a repetitive feature, it can be extracted computationally, it can be averaged, and then it can be put back precisely into context. That is now standard procedure in tomography, referred to as subtomogram averaging. Then we wanted to transition, of course, to cells. That was the goal from the beginning. And what you see here is we began to grow all sorts of cells directly on EM grid. What you see here are dictyostelium cells crawling on an EM grid. We were able about, almost 25 years ago to obtain the first tomograms of a cellular landscape by cryotomography. That is basically a peripheral region of a dictyostelium cell. You see the actin network and nestled in between with relatively high contrast ribosomes. But there was, of course, a problem. That means the problem was that only peripheral regions or appendices were thin enough to be electron transparent, at least to 300 kilovolt electrons. So we needed a way of thinning the sample without affecting its structural integrity. You can, in principle, do some sort of physical sectioning with a diamond knife, but amorphous ice or vitreous ice is highly compressible, and if you do that, you get compression of your sample of something up to 30–40%. That is, of course, not acceptable. So we put our resources, human and financial, into the development of an alternative technique, focused ion beam milling, using a dual beam instrument, an SEM, to monitor what we are doing and to monitor our sample, and then, a gallium beam in the beginning for ablating material to cut kind of non-compressed windows into the cell. Now, that method again is now widely practiced. You can use different geometries of ablating material. You can make that wedge-shaped, or what one normally does, and what is illustrated here at the top. You ablate material from above and below the area of interest or the volume of interest, and then you obtain a thin lamella, a slice through the cell, supported only by the surrounding ice. Now, a first application of that, we cut some lamella in the vicinity at the periphery of a nucleus of a HeLa cell grown on the grid. So the HeLa cell nucleus you see in the top left in color, and then on the right-hand side you see a thin lamella, which is typically something like 200 nanometers thick in the periphery here. Now we can take tomograms moving from inside the nucleus to the surface of the nucleus. So you see in the top region, we are in the region of heterochromatin. You see nucleosomes. They don't look very impressive, but if you average them, I think resolution goes easily to something like 8–9 angstroms. The real challenge, however, is to see the connectivity between nucleosomes, and that is still to some extent an unresolved issue. The path of DNA cannot easily be traced. So as you approach the inner membrane of the nuclear envelope, you come to the region where the nuclear lamina is, a network of very fine 3-4 nanometer thick intermediate filaments. And then, I mean, you get to the outer membrane of the nuclear envelope, and that is studied with ribosomes. Now we can do a couple of image processing techniques. You can do segmentation of the nuclear lamina, of actin and whatever. And when you have particles like ribosomes or the nuclear pore complex, you can do subtomogram averaging. Now what you see here as an average of the nuclear pore complex is not yet really impressive in terms of resolution, but it's remarkable because it's an average over only 4 or 5 copies of the nuclear pore complex. And the resolution to achieve in subtomogram averaging depends critically on the number of copies you can include into the averaging. It's important that we can push that further so that we can obtain a structure at least of that quality from a single nuclear pore complex. Now that is more or less already possible. Now, what you see here is basically a synopsis of all the previous data, either segmented or after subtomogram averaging. Now, I think I will talk a little bit about a different system, that is Chlamydomonas, which is a preferred system for us because the cytosol and also the nucleoplasma is less crowded than, let's say, in a yeast cell. So what you see here is a slice or a lamella covering basically going through the entire volume of a Chlamydomonas cell. Chlamydomonas has a second advantage, and that is that it has more or less an almost deterministic topology of its organelles. So it's predictable in which region, I mean, you will find which organelles. So you see here in the box, that is the region of the Golgi apparatus and the endoplasmic reticulum. So you must keep in mind that a cell is a large volume compared with the field of view of a tomogram. Now, so here I think you see in the movie, we go through that region. On the right-hand side is the nuclear envelope, and then where the arrow points to one nuclear pore complex, and you can do some averaging here, and you see resolution is here already quite a bit better because we have here something like 100 copies or so. Now, you can go on all sorts of particles. I mean, the easiest is always the ribosome because of its size and its relatively high contrast due to the RNA content. And here, what you see in this average is a ribosome sitting on the translocon on the SEC61 protein conducting channel. And if you look at that, you see that resolution is good enough to resolve individual alpha helices, indicating that resolution here is now in the range of something like 7 angstrom. Now, I will, in most of the following part of my talk, I will not talk about ribosomes, but the opponent of the ribosome, if you like, the proteasome. We have a long-standing interest in the structure and function of the proteasome. Now, before, however, going into in situ studies of the proteasome, I think I need to give a very brief introduction. Basically, we distinguish between the 20S proteasome in yellow here, that is the so-called core particle, that is where protein degradation takes place, that is the degradation machine. But in order to be functional in the eukaryotic system, it has to join forces with 1 or 2 regulatory particles, here in light blue. Their role is to prepare substrate for degradation. They recognize ubiquitinated substrates. They have ubiquitin receptors, and they can deubiquitinate, which is necessary in order to unfold then the substrate, and unfolding is necessary in order to transfer the substrate from the regulatory particle through a narrow gate into the core for degradation. Now, that is, I mean, a very old reconstruction, very low resolution, long before the resolution revolution, as people refer it, which was, I mean, quite a jump in improving resolution due to better technology. Now, we solved the structure of the core complex long time ago, almost 30 years ago, We went from eukaryotic systems to an archaeon, which had a more tractable core complex, because at that time, I mean, even the subunit composition, the stoichiometry of the eukaryotic 20S core complex was highly controversial. Now, that we could also make recombinantly, so we had almost unlimited quantities of material for crystallization, and we solved the structure by X-ray crystallography. You see on the right-hand side, on the top, you see basically in the core particle in the middle, there's this chamber. That is the chamber that is basically where protein degradation takes place, where the active sites are located. A very usual and long, enigmatic and controversial active site. Now it's clear. It is a single residue active site. It is only the N-terminal threonine that does the proteolysis. That enables very easily to develop inhibitors simply by extending the N-terminals by 2 residues. Now, that is an early structure about 10, 12 years ago of the 26S complex. Still, resolution was not good enough at the time to interpret this 8-9 angstrom map in terms of the subunit architecture. It is a complex subunit architecture, 34 different subunits, plus a couple of associated subunits, which are not integral part. Now, I think after the resolution revolution, we got a structure now. This is the human 26S proteasome at a resolution of 3.9 angstrom. Now, I think I must still continue ex situ for a moment to introduce you to the dynamics of the molecule, to the conformational landscape, because there are large motions of subunits in the process of degrading a protein. And they are of such a magnitude that even at the lower resolution in tomography, we can infer from the conformation what the activity status of a proteasome is in the cell. So we have the core complex, then there's a module of AAA subunits, and their role is basically to do the unfolding and the translocation across the gate and gate opening into the core complex. Now, I think in the following, we return now to the in situ situation. We study 26S proteasomes in cellular environments, and we monitor the cellular dynamics and response to metabolic shifts. So what you see in the column in the middle on the left-hand side is basically the locations of the proteasomes in the intact cell. There are 2 major locations. One is, lining the inner membrane of the nucleus. The nucleus is an important degradation compartment. And besides, in the area where the Golgi and the endoplasmic reticulum is, you see these 2 puncta. You can see them, and that is, the location, I think, of the ERAT system. I think you will hear much more about that, I think, later this week from Peter Walter. We are looking here at it from a completely different perspective. I think ERAT is extremely well studied, but how it looked in terms of structural in situ, I think that was unknown. Very briefly, proteins are synthesized on the ER. They are translocated into the ER where they are supposed to fold properly with the assistance of molecular chaperones. But then can go wrong. It easily goes wrong under stress conditions. And then I think we have misfolded proteins which cannot easily be removed and degraded in the ER. So in the ERAT system, they are retrotranslocated into the cytosol for ubiquitination and degradation. So we take a closer look. These spots here are highly dynamic. You see processes, de novo assembly, you see fusions, etc. So now we zoom in precisely on those by cryo-electron tomography. In the background, the light blue particles are all ribosomes. The dark blue are ribosomes sitting on a translocon. And the microcompartments where we find the proteasomes in red, they're always completely devoid of ribosomes. That there is very clearly a phase separation process at one point. Now, we zoom into one of these clusters or microcompartments, and then do a molecule by molecule analysis by image analysis. We don't do template matching here. We pick all the densities, then we do a classification of all the densities according to similarity. We obtain classes. We can do separate averages of the classes, and we can map them back very precisely. We know what the origin of the molecules is that contributed to the average. So we zoom in into one cluster here, sitting on the ER. Again, first the ribosomes show up, then we get into the cluster of typically something like 20 to 30 in red, 26 as proteasomes. There's a second component that is known to be an element of the ERAT system, that is CDC48. Again, I think we can do subtomogram averaging without completely unbiased, without any reference, and it converges very easily to a structure. And the number we find here compares very well with, global proteomics measurements. Now, I think we can then look what the activity status is, inferring that from the conformational status. And it's very clear, if you look at them, dependent on the distance from the membrane of the endoplasmic reticulum, that those, close to the ER membrane have, a high percentage of being in a degradation active status. And if we take those molecules and on the top right, do an average, we see, they are linked or they are associated with a not very well-defined density, which is not accounted for by the proteasome itself. So we concluded this is likely to be substrate, and it's not a single substrate, so it's a mixture of substrates here. So basically, that summarizes the findings here. To some surprise, CDC48 is not where we expected it, in close vicinity of the membrane. It's more at the periphery of those clusters here. So we concluded from that there might be a mechanism in which not CDC48 is the main element in translocating the substrate from the ER to the outside, but that the proteasome itself is involved in that. And as a matter of fact, there had been earlier work from the late Stefan Jentsch and from Michael Glickmans saying that translocation and degradation are coupled. Now we move for a moment to those proteasomes in the nucleus. And what you see here on the right-hand side, we are in the cytosol. On the left-hand side, we are in the nucleus. You see the nuclear envelope. You see nuclear pore complexes. We see in light blue, this were all the proteasomes, and then we can distinguish those between having one or two regulatory particles. We can distinguish between those, attaching or interacting with different structures, and we can distinguish between those who are in a ground state or which are actively degrading substrate. So many of them cluster on the nuclear pore complex, and there are, if we do a deeper classification, there are two populations. The red population, that is proteasomes that are tethered to the membrane at the periphery of the nuclear pore complex, and in yellow is a population that is associated with the nuclear basket. Now, I don't want to go into more detail here. I think a little story in between is from projects we have done, a collaboration between my colleagues Ulrich Hartl and Matthias Mann. So our interest was, on neurotoxic aggregates inside cells. And our interest was, what is the structure and the organization of neurotoxic aggregates in various disease states, and what is the interaction with the cellular environment. So the first one is, Huntington. So a terrible disease, characterized by a polyQ, a polyglutamine expansion. Then the protein becomes unstable and it forms an aggregate. Now, that is the structure of Huntington, which we did by single particle analysis. It's anyway a highly flexible molecule. You need to find ligands stabilizing it. Now, I think it is a bit challenging. I mean, in a neuron, it's a large landscape to target really in the preparation a Huntington aggregate. That is so we grow the neurons directly on the grid, then we identify cells which include and carry, the neurotoxic aggregate, then we cut a lamella, as you see on the top, on the right-hand side, and then we can do tomography of such a Huntington aggregate in situ. You see in blue the fibrils of Huntington, and in red you see remnants of the endoplasmic reticulum. So whenever a Huntington fibril touches upon the membrane of the ER, it changes dramatically its curvature locally, and that leads to disruption of the ER, and the ribosomes in green become all liberated. The next one is the situation of ALS, which is characterized by hexanucleotide expansion that leads to the synthesis of toxic D-peptide repeats, and in patients suffering from that disease, we find typically glycine-alanine repeats. We do the same experiment here. It looks at a glance maybe similar, but these are not fibrils, these are polymorphic ribbons. And these polymorphic ribbons are inhabited by macromolecules. We do again a molecule by molecule analysis, and you see basically there's only one molecular species inside the aggregate material in light green, and this is 26S proteasomes. Ribosomes cannot enter the aggregate material, and the same is true for the chaperonin trick. Now, since we have many copies of the 26S proteasome there, because locally the concentration of proteasomes becomes enriched something like 30 to 44-fold inside the aggregate material. On the right-hand side in blue, we have proteasomes which are in the substrate processing state. And again, we see there always one mass in pink associated, again, not accounted for by the proteasome structure itself. So we concluded that is again substrate interacting, but we can test that. We map the blue ones all back to the tomogram, and invariably they are in direct physical contact with the ribbons. Now that is from a News and Views written by Wade Harper comparing the two studies I just showed to you. I think in the last part of in situ, the studies of the proteasome, a very new study. Now it is clear that, proteasomes are great consumers of ATP. And so the question was, what does the cell do under conditions of shortage of ATP, so under conditions of starvation? Now, it was known that under conditions of starvation, the proteasomes leave the nucleus. That was known from light microscopy, as shown here, and they accumulate in puncta, which are referred to as proteasome storage granules. So the question that came up immediately, what is the structure of proteasome storage granules? It was known that proteasome storage granules, the proteasomes there are proteolytically inactive, and the explanation was what is indicated on this cartoon, that the core particle and the regulatory particle had fallen apart from each other, and therefore they could not degrade. So we decided to follow basically the dynamics along the proteasome storage granule formation pathway. And there was a very surprising phenomenon at the onset of starvation in the nucleus. The 26S complexes form or assemble further into trimers of the 26S2 complex. And you will see in a minute that, when they form, that is the structure here at something like 7-8 angstroms of the proteasome trimer, they interact via the regulatory particles and they become arrested in the proteolytically inactive ground state. A very convenient mechanism of shutting them down just by assembling further. Now, the 26S proteasome trimers then, they congregate at the nuclear envelope in the vicinity of nuclear pore complexes, ready for leaving for leaving the nucleus. Now then they move into a condensate that is initially liquid-like, but then solidifies over time. And it solidifies because in this condensate, the trimers assemble further. First, they get stacked in the form of fibers, and the fibers then, align laterally with each other and form then bundles. So basically, they form a very large paracrystalline array in these condensates. That is the reason why. And the structure, of course, of the trimer in the granule, in the proteasome storage granule, is exactly the same as the trimer in the nucleus. Now, I think I shortened that a little bit. So when you do FREP or other photobleaching experiments, you can see how it solidifies as the paracrystals grow. Now, one last thing here, I think we looked more closely into the interaction in the trimer, and it's pretty clear that the main subunit that is at the interface between the complexes is RPN9, one of the subunits of the regulatory particle. So RPN9, we decided then to modify that. We did some mutagenesis. We removed or shortened the N-terminus. In all those cases, what happens is that then the proteasomes remain in the nucleus. They cannot leave the nucleus. And as you see also on the bottom right, they remain monomeric in the nucleus and remain there. Now, what happens in the granules with the paracrystals, if you replenish, the medium, then I think ATP can be produced again, and then many or some of the proteasomes adopt non-ground state conformations. And then, if a few of those, adopt a different non-ground state conformation, then the whole thing kind of collapses. And then it means basically that proteasome storage granules are a readily releasable pool of fully assembled 26S proteasomes, which are then imported back into the nucleus. I think the last few words, that is from a review I wrote with my former graduate student, Martin Beck. Can we, in the end, go to real atomic resolution? And I think there's not, a couple of things need to be improved. We need to improve throughput, which is currently not limited by data acquisition. Taking tomograms has become quite fast. You can easily take now on one machine 300 tomograms per day. We need to improve the primary data quality to some extent. Cameras have been massively improved in recent years, but they're still not at the limit, not with the perfect detective quantum efficiency. And I think a big hope in the field is the laser faceplate developed by Holger Müller and Bob Glaser in Berkeley to boost contrast. We must also increase the coverage of cellular space. You must keep in mind that a thin lamella accommodates basically only maybe 1% or even less of the volume of a cell. So we look at a relatively small subvolume. Now that can be improved quite dramatically by serial lift-out we have developed in recent years, so we can bring it up to something like 30%. I think the biggest challenge is improving data interpretation. Basically, the image we have, it's a label-free method, and so what we image is the spatial modulation of density inside the sample. And I think the method to interpret that, to identify and annotate all the densities in a tomogram, and there are millions or billions of densities in one tomogram, will be done by something like template matching. To illustrate that briefly with an experiment which we have done together with Axel Brünger's lab in Stanford, what you see there is he's interested in synaptic structures, especially in synaptic vesicles. You see a part of synaptic vesicle. The large structure is the V-ATPase. Subtomograph averaging gives a very good structure, can easily be recognized, but there is a small density in kind of this orange, brownish, and so-called extra density. So the question was, what is this? What is the identity? And the strategy we have used here was basically, there was a limited number of candidates from the size, only 4, synaptophysin, synaptoporin, and synaptogyrine. What we have done is we have generated AlphaFold models and compared their similarity to the observed density. And minimizing outliers, I mean, we get a measure, we get a similarity score. So it was very clear that synaptophysin was by far the best fit, and we could then validate that by knocking out synaptophysin, and then the density disappeared. So a few words about the next frontiers. I think the next frontier is not just to observe, to focus on one or a few molecules in the cell, but to do it more globally by something we call visual proteomics. I mean, the tomogram is a fairly comprehensive visualization already of the entire proteome, but, well, the problem, as I mentioned before, is of course still identification and annotation. Now, once we achieve that and we scale the template matching method, then I think we can create whole cell computational models or digital twins, which allow us to do simulations of phenomena which are difficult to test or to do experimentally. I think finally, just with a quotation, I think new directions in science are launched by new tools more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have to be explained. So that is basically, very briefly, only the acknowledgement of my colleagues in the past 3 or 4 years. Thank you very much for your attention.
Thank you. Thank you so much, Professor Baumeister, for that truly inspiring and forward-looking presentation. Thank you. Ladies and gentlemen, we'll now be taking a 30-minute short break. Refreshments will be available at the back of the hall. Please be seated back here at 11:15, and we'll have our next panel discussion session. We'll see you later. Thank you.